Georg Fuellen, thank you so much for being here. Professor at the University of Rostock, Bioinformatics and Aging Research. Thank you for being here.
Yeah, thanks Aubrey for the invitation. I try to do it in 20 minutes so that I can have maybe some questions from the audience. It's about effective, safe and personalized health interventions. I'm touching 4 kilograms of strawberries and some bioinformatics. And the key question is how to intervene in a safe, effective, and personalized manner. And especially safety and efficacy is kind of a trade-off. The safer, the less effective usually, or the more effective, the lower the safety. And one idea is actually to use a digestive system as a safety net. And also known as trying out nutritional interventions. And in this talk, I would actually like to focus in particular on maximizing the effects of a polyphenol-rich diet. And I will show some evidence that polyphenols can kill senescent immune cells by diet, that polyphenols can rejuvenate the epigenome. That's an idea. We are currently measuring some of that. And I will show how artificial intelligence systems can also generate recommendations regarding healthy diet by suggesting reprogramming mimetics. And finally, on 2 slides, I will talk about how polyphenols may help the climate and health. Yeah, nutrition for longevity. So I talked about safety net and also personalization. That's by food aversion, by vomiting, diarrhea. But it's not just that in case of nutrition. You also have the microbiome, which is improving, often is improving bioavailability. It's producing butyrate, for example, short-chain fatty acid, which is good for brain health. And with food, you are also expecting lots of synergy among the different ingredients. So it's not about isolated compounds like quercetin only or vitamin E only. It's about compounds in synergy. But there are also severe downsides. Some of them is actually low effect sizes, and I really want to do something about that. There's not much standardization. So, of course, the food is very variable by day, by week, by season, and so on. And it's really hard to study. There's the issue of placebo effects and some other issues. But maybe with bioinformatics, and that's also in the second part of my talk, you can maybe maximize efficacy. You can maybe learn from high-risk interventions. You can look at similarity in biomarker space and try out nutrition interventions that are somehow changing biomarkers in the same way as high-risk interventions. And also the other way around, bioinformatics for maximum safety. You can look at high-risk interventions and you can try to de-risk them by looking at similarity to nutrition interventions. But now about testing the limits of what nutrition can do, we designed a study a few years ago, which is centered around polyphenols. Polyphenols is a colorful stuff in plants. It's also used by plants for pest protection. And there are lots of papers that they can improve human health. And one polyphenol you might know about is fisetin. It's said to be in strawberries in large amounts. We're actually not sure in hindsight. Then another one is quercetin. It's in capers in particular. The largest amounts are reported to be in capers. And this is actually sweet and salty food, so it's a kind of complementary food. And we were worried about all the sugar in strawberries, but when you look closer, actually strawberry powder is used to prevent and also to treat metabolic syndrome. So it's probably not so much of a problem, maybe because the fibers are also enabling a better metabolism of sugar. And in the trial, we then use fresh and dried strawberries to maximize the polyphenol intake. And we hope lots of synergies there between fisetin, quercetin, also anthocyanins. Actually, pelargonidine is an anthocyanin giving the red color to strawberries, and there are also catechins. And the study was conducted with Michael Walter, Alex Hartmann from the Laboratory Medicine in Rostock. And you can think of it as a fortification of the Mediterranean diet. And that's a study design in that we had 170 healthy elderly people recruited into 5 groups. You can see group 1 up to group 5. And they got more and more in terms of polyphenol-rich food. And it went on for 10 weeks for a participant. And for group 1, which was kind of control group, they just got 500 grams every week, just to keep them happy. They got it for free from a Colorado strawberry farm. And group 2, we actually escalated the dose. So it went up from 1,500 grams to up to almost 4 kilograms in the last week. And then for groups 3 and 4 and 5, we added dried strawberries, but only on certain days. So the first day of the study, they had to eat 100 grams of dried strawberries, and then another 100 in group 3, and also at the end. But in group 4, we added more dried strawberries. And group 5 then was a group with the highest amount of polyphenols. We added capers and olive oil, actually with olive oil, so they were also supposed to eat the olive oil with the capers. And in the end, what people had to eat is visualized on the next slide. So it's dried strawberries, 200 grams, which is about 2.5 to 3 kilograms of fresh strawberries, then 750 grams of fresh strawberries, original fresh strawberries, and 240 grams of capers and olive oil. And of course, you don't want to eat that every day. It's quite a lot. It was on 3 single days over those 10 weeks. And actually, the digestive safety net is not perfect. The amount of quercetin in here is so large that you may actually endanger yourself if you are also taking heart medication. It's competing for the same enzymes. You may know about grapefruit juice, which could be pretty dangerous. And here we are also not recommending this to people who are taking digoxin, for example. Okay, and what did we find? So here, it's all in comparison to the control group, group 1. The first row is group 2. And an arrow, if it's dashed, it's just a trend. And if it's a thick green arrows, those are significant changes. So for group 2, already with the fresh strawberries, you could see that LDL cholesterol went down if it was high at baseline. And then we did gene expression data and peripheral blood mononuclear cells. And here we found an upregulation of the aerobic electron transport chain. And so that means better cellular respiration, better functioning mitochondria. And we got a downregulation of inflammation. For group 3, in a significant way, we found that C-active protein went down if it was high at baseline. For group 4, the total cholesterol got was downregulated significantly. And for group 5, it's actually the most interesting. On the one hand, LDL cholesterol, the downregulation got significant, whatever the baseline value was. And we also had better respiration. But then we found apoptotic signaling going up. But inflammation still staying the same. That's quite interesting. And our hypothesis is that senescent immune cells were killed. And this killing actually then causes some inflammation. So the inflammation no longer went down, because this was a group with the highest polyphenol food on the day just before the last blood draw. I actually forgot to mention this. Maybe here I can go back once. Whatever you have as a thick line is a blood draw. So the blood draws were done at the very beginning and at the very end, and also in the middle. And the last blood draw was just done after the last big seno intervention. Okay, and yeah, and LDL and CRP, they were pre-specified as primary endpoints in the ESSIS application. The analysis were done by Henrik Rudolph and Ricardo Secchi. Yeah, this is a gene expression data done by Ricardo. And in more detail, for the group 5, with the highest intervention compared to control, baseline versus last day, as I said, we found better respiration, and we found cell killing and positive regulation of the apoptotic signaling pathway. And the question is, what are the compounds, what are the ingredients in the food that may do that? I already mentioned fisetin. There was a paper some years ago actually claiming that there are large amounts of fisetin in strawberries. And there was also a paper then by Robbins, Niedernhofer, and others that fisetin is a xenotherapeutic, that it extends health and lifespan actually in mice. That was actually the motivation for the study. And here you see fisetin in a mass spectrometry plot. However, that's from the smoke tree control sample. And we did not find any fisetin in strawberries. You find it all over the internet, but we didn't find any. But what we found is actually lots of quercetin, even more than reported previously. And it's in the fresh and also in the dried strawberries. And now, of course, also those color pigments like anthocyanins. Right, and quercetin is actually not so different from fisetin. It's just one small OH group. So maybe actually fisetin gets interconverted to quercetin, maybe even in the strawberries. Who knows? That was done by Ritu Schwarz, Burkhard Hinz. So our lead is actually now quercetin, if you want to explain what happened. And this fits very nicely, because quercetin is known to induce apoptosis in vitro and in vivo in animals, actually by inhibiting histone deacetylation and by inhibiting DNA methylation. And since there's so much quercetin in strawberries and capers, our hypothesis is that it's causing senolysis of senescent immune cells. But we still have to measure a few markers for that. Right now, we can just say it's fitting. It's a good hypothesis. And it also fits with a high amount of inflammation in the last group, which is kind of counteracting the general downregulation of inflammation. And we went for xenointervention days, just a few, also because senescent cells, you don't need to kill them every day. They come back within weeks or months, or at least within a longer time period. And we also know that there's a lot of rejuvenation of the epigenome going on in vitro, and we also want to still measure this in our study. And this is nicely matching actually this chemical reprogramming. You probably all know about Yamanaka factors, and maybe also that they can be replaced by chemical reprogramming cocktails. And for the first steps, especially some molecules are used that are also epigenetic modifiers, like valproic acid and butyrate, which inhibit histone deacetylation, or 5-alpha-DZ and decitabine, which inhibit DNA methylation. So we are not curating data. We've got a tabulation of transcriptomics data describing transient or partial reprogramming. This is a table. You are not supposed to decipher it, but it's just giving you an impression. We did a lot of work on getting a hold of also of the data sets and listing all the biological findings in there. And one thing we now want to do is we want to compare that human data curation with what large language models can do. And we want to use the data for biomarker similarity analysis. And on that, comparing data in biomarker similarity space, we've got some first pilot results. This is actually a PCA plot. So the plane is a similarity space. And what you have in green are low-risk nutritional interventions. Actually, 2 studies, and the interventions are very, very much alike here. That's why I clustered closely together. And also high-risk interventions, essentially transient reprogramming, which are a bit more heterogeneous. And in this similarity space, based on gene expression data, you could say that the high-risk but safer interventions may be the ones that are closer to the lowest interventions, just because of the gene expression data. So this is a bioinformatics for maximum safety, trying to de-risk high-risk interventions. You could also do it the other way around, as I already said, but this is not really in this plot because the lowest interventions are, they are not closer or farther away from the high-risk ones. This was done by Daniel Palmer. And now in the second part, actually, we are looking for a high-risk intervention of Parnate and Rapsox. Those are 2 chemical compounds which are used for chemical reprogramming. And last year, there was a preprint by Ocampo Lab, and they specifically looked at reprogramming as it ameliorates hallmarks of aging and extending lifespan in cells in particular. And they've looked at the combination of TCP, which is also known as Parnate, and Rapsox, which is actually improving DNA damage markers and also cellular senescence markers. And now the question is, are there mimetics for Parnate and for Rapsox, nutraceuticals, or even food? And I asked a large language model, actually in 2 steps. the first step, so I used plot 2, and I asked it what Repsols is actually doing. So I started to describe that Repsol is a chemical compound and so on, and I wanted the model to describe its general effects, sorted by, especially by in vitro, animal, and potential human effects. And then it actually produced a nice summary. It started as that it's inhibiting TGF-beta, which is correct, and also that it's helping with IPS generation. And there are actually also some health effects, like in mouse models for Alzheimer's disease, but no human data yet. I also asked it about Parnate. And now the interesting part is, I said, no, you now listed, you just listed about the effects of Repsols and Parnate to come up with Nutraceuticals that are reported to have closely similar effects. It actually did that. So it said some general statements, then it listed curcumin, resveratrol, nicotinamide riboside, and quercetin. So quite nice. And I then asked it to do the same listing, but instead of nutraceuticals, taking food items and also adding some references. Yeah, and very nicely it said, okay, epigenetic modulation is there with broccoli, okay, and berries. Pretty nice. And even it mentioned olive oil, which we also had in our study that I showed you. It also produced references, which were actually correct. And I asked it for a dietary recommendation in general. So what kind of diet enables actually a mimicking of Repsols and Parnate? And it said, okay, broccoli, berries, green tea, veggies, citrus fruit, and then extra virgin olive oil, green tea, and blueberries. No strawberries, unfortunately, but okay. And this is references for those. And actually, if I go back, what it also did, I didn't mention that, it was able to say for those processes whether Parnate or Repsols is responsible. And now if I move forward again, it also did something similar for the nutritional compounds. Yeah, and then of course I couldn't resist to ask it for some slides. So this is a kind of summary of the investigations I did with Claude 2, discussing potential anti-aging effects of pharmaceutical compounds like Repsols and Parnate, reviewing possible natural equivalents in foods that may mimic these effects. I looked at limited evidence for other compounds also. I asked it, for example, for valproic acid, for scoline and others. And the goal was dietary recommendations for lifespan extension and rejuvenation. And then a specific slide on berries, potential anti-aging effects of berries. So berries contain anthocyanins, but also of course quercetin that act as HGAC inhibitors, similar to Repsols. Berries with resveratrol activate the CYP1 pathway, like effects of Parnate. And polyphenols in berries provide neuroprotective benefits seen with Repsols and Parnate. And also some cognition improvement of cognition. Compounds in berries mimic those pharmaceutical anti-aging effects on a smaller scale, but human data are still needed, and berry consumption is probably good for anti-aging. The key takeaways, so it's about understanding cellular pathways, and then this mimicking of effects and modulation of aging mechanisms. More research is needed, and in particular, last point, synergy between foods is also very important. Okay, now towards the end, we did a first study actually in December 2019. That was just me. And then I convinced 3 colleagues to do it, and then we had an official pilot, and then the study that I presented. And what I found out is actually a rise in body temperature. Maybe it's just me, I don't know. So we really like research on the influence of healthy food on body temperature, especially after eating and also in the longer term, over the next days. And it's so easy to measure. I couldn't convince my colleagues, and it's really rare to find such data. But it's small effects which could be working on a large scale. They could really be relevant. And one example just is green tea. It activates BAT, bone adipose tissue, increasing metabolic rate, non-shivering thermogenesis. And that actually then catches two birds with one stone. Namely, on the one hand, you are happy because you are feeling more comfortable, you have got healthy food, and you can also save on the energy costs. And in summer, resveratrol as a characteristic mimetic may actually do the opposite. That was worked together with Axel Kowald. And as a summary, so intervention days may kill senescent immune cells, polyphenol-rich diets improve blood parameters, and especially you have got anti-inflammatory effects and better lipid profiles. There's no quercetin in strawberries. Probably not. It's always hard to prove that something is not there, but lots of quercetin. And we hope that nutraceutical mimetics can rejuvenate by reprogramming. And next winter, green tea keeps you warmer. And we published a strawberry study as a preprint, and there's also this review on extreme temperatures. And finally, I'm looking for a postdoc in 2024. I'm looking for people who are interested in those topics. Please contact me. I'm welcoming you to Mecklenburg-Vorpommern, of course. I'm saying the team, the partners, the participants, and the strawberry farm who funded some of the food. Thank you very much.